Okay, so this is the urinary system, the beginning of our discussion about the urinary system, that is. In this lesson, you will learn about the major organs, you'll learn about the nephrons, and we are going to begin the discussion of the physiology of the urinary system. Specifically, how filtrate is made and modified within the nephron. Okay, so this really does set the stage for the rest of our discussions on the urinary system. Okay. So as always, let's first begin with the functions of the urinary system itself. The urinary system perhaps is underappreciated. You think it's just making urine, right? Just getting that extra waste out of your body. And that, of course, is an important function of the urinary system. We do remove toxins. We do remove metabolic wastes like urea, uric acid in the urine. But it's so much more than that, right? There are so many extra factors that we can regulate using our urinary system. So you should think about the urinary system as filtering and regulating what's happening in the blood, right, or what is contained within the blood. Remember, the blood is highly regulated as opposed to the lymph, which just has everything in it that is in the surrounding tissue. Blood, we need to maintain levels of ions, of nutrients, of cells, all these different things within a very narrow range. And so we begun our semester really talking about how important it is to maintain homeostasis via the blood and how the blood has to be highly regulated. This is one of the main ways that we do it, right? This is really the main organ system that is filtering the blood and regulating everything within it. Okay, so we've talked about a lot of other types of filters, such as the liver, such as the spleen, but this is the big one. Your kidneys are so important for regulating the ion contents, right? So how much bicarbonate, how much hydrogen, right? And of course, that leads into the pH. Right, so this is the final piece of that pH balance story that we started talking about again the very first day that we spent together this semester. So the kidneys work together with the lungs, work together with the blood in order to maintain a pH of 7.4 in your blood. Okay. Also, as we begun to talk about in the first section of this class, the urinary system functions in regulating blood pressure. Remember that renin, angiotensin, aldosterone system is going to work at the level of the kidney to either keep water in the blood and therefore increase blood pressure, or kick a lot of water out and therefore decrease blood pressure. Okay, so we talked about the hormones before. We talked about stretch receptors and whatnot that actually lead to shifts in blood pressure. But now, again, we get the last piece of this story. Hopefully the entire semester starts to come together in this particular section, because now we get to see how the nephron, which we'll start to learn about today, can literally select, we want these ions, we want this water in or out of the body. Okay, we regulate nutrients. So for example, our blood glucose levels. Okay, so we'll talk about diabetes, and we'll connect diabetes back to what's happening in the liver. And of course, looking at urinalysis tests, right? So what is in the urine and how we can look at urine to figure out what's going on in a lot of the other organs of the body. Okay. The kidneys also have endocrine functions. And again, we've talked about this a little bit before, but now we really bring all of these things together. The kidneys make renin to regulate blood pressure, and the kidneys make EPO to regulate blood cell content in our within our plasma. And finally, the activation of vitamin D. Remember that calcitriol is a derivative of vitamin D. And the kidney is going to help activate this particular vitamin so that we can absorb calcium from our diet. So absorbing it across that mucosal barrier of the digestive system and into our blood. And the kidney is going to make sure that we have just enough of that calcium. And so lots of different functions. Really, the urinary system isn't just making urine. That's just kind of a byproduct of its real functions, and that is regulating the blood. Okay. The primary organs, primary functional organs of the urinary system are the kidneys. Your kidneys are pretty much plastered all the way up against the back of your abdominopelvic cavity. Here we can see the posterior side of the body. Down here at T12 to about L3, we have the kidneys, and they are actually retroperitoneal. So I believe we've seen this transverse section of the abdominal cavity in the past. Here's the liver, here's the stomach and the spleen, and we can see that all of those organs are outlined in pink, and then they are suspended within this cavity that's outlined in like tealish blue. And so all of those organs are surrounded by the peritoneum. Okay, so this membrane that kind of allows all of these organs to shift around without rubbing up against each other to hold them in place. However, the kidneys, as we can see, are outside of this peritoneum, right? We can see that they're just kind of like suspended right here next to the spine. And so they need a little bit of a different type of technology, a little bit of a different type of structure to actually keep them suspended there, right? There's not like ligaments that they're hanging by or anything like that. There's no peritoneum to keep them up. Instead, they are held in place by a series of things. First of all, there is a fibrous tissue capsule. Okay, so if we were doing a dissection, we would see, after we took the entire digestive system out of the cat, we would see that the kidneys are actually like fused to the back of your body cavity, really, by this really thick fibrous material. Within that that fibrous tissue capsule, we have a lot of fat, right? And this is not something that you're going to go to the gym to get rid of. In fact, it's really bad if you are so malnourished, right? You have been dieting so intensely, or there's physically not enough food available to you, that if you start to lose fat around your kidney, essentially what that does is it allows the kidneys to drop down farther within this capsule. Okay, what that can do, right, when your kidneys start to descend here, is that it can actually pinch off or kink the ureter or ureter that is taking urine from the kidneys and out into your urinary bladder. Right, so that's really bad. In order to keep filtering your blood and maintaining homeostasis in that way, urine actually has to be able to leave the kidney. And so if you do not have enough fat, right, fat is not this evil thing, we need a certain level of fat, then your kidneys will descend and pinch off that ureter. Okay. So again, the kidney's main job is to regulate what is within the blood. And so how much blood are the kidneys getting? Well, the renal arteries, which again, we talked about long ago, weeks, months ago at this point, the renal arteries deliver about a quarter of the cardiac output. So the entire volume of your blood to the kidneys every single minute, right? So every 4 minutes, you are filtering the entire volume of your blood. Right, so the kidneys are super important and super active in this process. This here, of course, is a section through the kidney. We can see the renal arteries in red delivering oxygenated blood essentially to this area right here. Okay. As you will learn in the lab, the kidney is divided into essentially the outside structures and the inner structures. And so just to give you a couple words right now, the cortex, right? Just like the cerebral cortex is on the outside of our brain, the adrenal cortex is on the outside of our adrenal glands, right? Cortex means like on the outside. And so the renal cortex is this lighter colored tissue out here. The medulla, right? Medulla oblongata is like kind of at the bottom in the middle of the brain. The adrenal medulla is all the way in the middle of the adrenal gland. And so the renal, right, meaning kidney, medulla is this middle section in here. And so this is actually broken down into what are called renal pyramids. Again, you'll learn more about that in the laboratory. But these here are renal pyramids, and collectively, this is the renal medulla. And so I bring this up to you now because oxygenated blood is delivered via these arteries, via these blood vessels, to the boundary between the cortex and the medulla. And the differences in the tissue composition and of that interstitial fluid, the fluid that's just kind of like hanging out outside our cells, is going to be really important in this process. Okay, so again, we can see that the blood is delivered to this, what is called Corticomedulary Boundary, right? It's the boundary between the cortex, the outside, and the medulla, the inside. Okay, if we were to zoom in on that boundary, what we would see is something that looks like this. Okay, right? The red vessels are delivering oxygenated blood. The blue vessels are draining deoxygenated blood. And in the middle there, we have the passage of blood through not only one capillary bed, but two. And so here today, we get to talk about the third and final portal system within the body. Okay, the first one we talked about was the hypothalamus and anterior pituitary gland, right, passing through two different capillary beds before returning to the heart. The hepatic portal system, right? So blood passes through the capillaries within the digestive system and then passes through the sinusoid capillaries within the liver lobules before returning to the right atrium. Okay, so this is the third and final portal system. Okay, as we can see, these red vessels deliver blood to this capillary, right, via an arterial. Now, the vessel that is delivering the blood to this first capillary bed itself is called the afferent arterial. Okay, so A meaning going to, right, approaching this first capillary bed. Okay, the first capillary bed itself is called the glomerulus, right? Sometimes it's called glomerular capillaries. Lots of different words for the same structures in this particular section. Okay, so the afferent arterial is going to deliver blood to the glomerulus, right? The first capillary bed. Right? Some magic filtration stuff is going to happen. We'll get into that here in a moment. And then some of the blood, right, less blood in fact, is going to drain out of the capillary bed and into a second capillary bed. Okay? And so the vessel that's draining this capillary bed would normally be called a venule, right? Because venules drain the deoxygenated blood. However, we can see that the blood here is still red. It still has oxygen. It hasn't actually unloaded oxygen at all to these tissues. And so we call this the efferent arterial. Okay, efferent for exiting, it's going away. What is it going away from? It's going away from the glomerulus. Okay, so just a little bit of a different terminology here than we're used to. Afferent arterial going to the first capillary bed, efferent arterial leaving the first capillary bed and delivering blood to the second capillary bed. And so we can see that the second capillary bed is much larger and it actually covers and kind of weaves around this funny little yellow tube here. All right, we're going to spend lots of time talking about the yellow tube. The yellow tube is collectively called the nephron, and the capillaries that surround and are exchanging materials with this nephron are called the peritubular capillaries and the vasa recta. Right? So what's the difference here? Well, the peritubular capillaries are the capillaries around the nephron within the renal cortex. Right? So within this outermost region here. These are peritubular capillaries. The vasa recta, right? So rectus, rectum meaning straight up and down. Vasa meaning these blood vessels. So these straight up and down blood vessels. And so we can see that there's actually a portion of the nephron that's kind of straight up and down. And so the vessels that are exchanging materials with that straight up and down portion are called the vasa recta. And again, all of this is happening right at that boundary between the outer portion and the inner portion of the kidney, the corticomedullary boundary. In fact, these straight vessels here are literally right along that edge. Okay, so this is in the cortex, this is in the medulla. Okay, any questions at this point? All right. Okay, so here's another perspective. Here is a renal pyramid, which is part of the medulla. This is the cortex. And again, these squiggly guys here are the nephrons. And the blood vessels, of course, are in red. Here's the glomerulus. These are the peritubular capillaries and the vasa recta. Okay. The nephron itself is the functional unit of the kidney. Right? So when I talk about all of these different functions of the kidney and how the kidney's job is to make sure we have enough red blood cells and we have enough glucose and we have enough salt, right? All of these different things. That's the kidney's job. But when we say the kidney, we're actually talking about the nephron. Okay. So the functional unit of the kidney, we have over 1 million of these things in each one of our kidneys. So over 2 million nephrons give rise to the functions of the urinary system. We can break the nephron down into 2 main parts. The first part is the renal corpuscle. Right? The renal corpuscle is, first of all, where those capillaries are, the first capillaries. Right? So the glomerulus, this knot of capillaries, is within the renal corpuscle. Okay. The second portion of the renal corpuscle is called either Bowman's capsule or the glomerular capsule. As always, we're trying to move away from using people's names to describe structures. And so the more current term, the more up-to-date term is glomerular capsule. However, in the past, I have found that glomerular capillaries versus glomerular capsule just gets to be so clunky and so confusing when I'm talking about both of these things that I am going to choose, for your sake, to say Bowman's capsule as opposed to glomerular capsule, right? You should learn both terms, right? Especially because as you progress in your medical career, you're going to start seeing more and more of these terms that don't use people's names. So you really need to learn the newer term. However, it's super clunky and super confusing. So I will say Bowman's capsule. Okay. So what is this thing in the first place? Well, the glomerulus, I'm going to keep representing by like a fist, right? It's literally a knot of capillaries. Right? So here is the glomerulus, and Bowman's capsule is the beginning of the nephron, right? And it actually wraps around the glomerulus, right? So it cups around the glomerulus, blood comes into the capillary bed, and what's going to happen there is that components of the blood, not all of the blood, but components of the blood are going to go from the glomerulus into kind of like this catcher's mitt of the Bowman's capsule, okay? All right, so that's the first part of the nephron. The second part of the nephron is essentially playing with that filtrate, right? So components of the blood came from the glomerulus into Bowman's capsule here, right? And that fluid and everything within that fluid is going to be called the filtrate, right? So it's not technically the blood. There aren't actually red blood cells, but it came from the blood, right? It's a filtrate. We filtered some stuff out of the blood, and now all of that stuff is going to pass through this squiggly little tube with the straight part and back up with another little squiggly part. All of those things are collectively called the renal tubule. Okay, so everything that is actually holding on to the filtrate, which will become our urine, is called the renal tubule. Okay, so again, the nephron contains the renal corpuscle, where filtrate is made, and the renal tubule, where filtrate is modified based on whatever our body needs at the time. Okay, there are several functions that occur, of course, over the entire length of this nephron. And first of all, we have glomerular filtration, right? So stuff is going from the blood into the catcher's mitt of the Bowman's capsule, right? Bowman's capsule is catching all of those things that we're going to filter out of the blood. And then we play with it, right? We reabsorb some stuff back into the blood, right? So from the filtrate, which is essentially going to become urine, but we don't want to just pee out all of that glucose that we just spent so much time digesting and absorbing and processing. We don't just want to pee it all out. And so we reabsorb glucose, for example, back into our body. Also, not everything that we don't want was actually filtered in the first place. Okay, so any extra toxins, any additional ions that are in excess of what we need, any more metabolic byproducts that just never got filtered the first time, we are going to actively secrete them into the filtrate, right? Dumping them from the blood, from the peritubular capillaries into the filtrate. And then ultimately the filtrate is going to be drained out this collecting duct and down to your urinary bladder. Okay, so that's the third and final step to this process here. We drain the urine, right, at this point into the collecting duct, right, which is right over here. Ultimately out the kidney, to the bladder, and out. Okay. So that is where we're going here. I want to elaborate just a little bit more on this filtration versus reabsorption versus secretion, because these are things that we're going to be talking about again and again and again. Okay, so the difference between these three terms. Filtration is something we've actually seen before. We've seen it when we talked about the capillaries long time ago, right? And so here is the capillary. This is where blood is. This is inside Bowman's capsule, right? And ultimately within the tubule. And what we see is that because of the blood pressure, certain things, certain solutes are going to be small enough to be physically pushed through that capillary membrane and pushed into Bowman's capsule, that catcher's mitt over here. And so the driving force behind filtration is the blood pressure. Okay, it's physically pushing stuff through this selectively permeable membrane. Okay, anything that's too big to pass through these pores is going to stay behind in the blood. Anything that is small enough is going to enter into Bowman's capsule. And this is an indiscriminate process. Pretty much if it is small enough to pass through, it's going to go into the filtrate. Okay, and that's why we spend the rest of the time in the renal tubule kind of playing with what is in that filtrate. Okay, so this is a filtrate. It has the same solute concentration as the blood. Okay, of course, we want to modify that, and so we reabsorb the stuff that we want. Okay, so here we can see some of those solutes, glucose, for example, we don't want to just pee out. And so we utilize transport proteins to bring the solutes that we want, the good ones that we need to maintain homeostasis, back into the body. All right, so this over here is the blood. It says peritubular fluid because technically these solutes are going from the tubule lumen into the space within the kidney, and then they're going to jump into the capillaries, right? So that's why it says peritubular fluid, peri around the perimeter, tubule, what? Around what? Well, the renal tubule. Okay, so reabsorption is taking back the stuff we want into the body out to the filtrate. Secretion is taking stuff that's left over in the blood that we really don't want to be in the blood anymore and pushing it or allowing it to diffuse, depending on what we're talking about, across that tubular membrane and into the tubular fluid. So pretty much by the time the filtrate gets all the way through the nephron and into the collecting duct, that's it. Whatever is left in the filtrate is going out with the wash. Okay, so secretion is going to add stuff that we don't want into that waste. Okay, in order to reabsorb and secrete, we have a series of membrane transport mechanisms that are going to facilitate this process. And so just as a reminder, passive diffusion is allowing substances to flow with their concentration gradient. All right, so what we can see here is that there's a high concentration of solutes in the filtrate. And if we stick a particular protein within the membrane of the tubule, these substances are just going to flow down their concentration gradient and back into the body. Okay, so that's passive diffusion. There's no energy associated with that. Okay, certain things can just jump over the membrane, right? So respiratory gases of course, can just pass over the membrane with their concentration gradient. Facilitated diffusion is much more common in the kidneys. Again, that is adding these transport proteins into different areas of the tubule to allow solutes to go with their concentration gradient, but only at certain parts, and we'll get into that in a moment. Okay, remember that osmosis is a type of facilitated passive diffusion. We never pump water, right? We never use energy to physically take water molecules and move them somewhere. We simply add aquaporins in different parts of the tubule to allow water to follow salt. Okay, and then the other type of transport mechanism that we use is, of course, active transport. Okay, so actually using ATP to pump substances across or against their concentration gradient. Sometimes we use secondary active transport, right? Just like we saw with the absorption of carbohydrates and proteins and nucleic acids, right? We create a concentration gradient of sodium and then use that sodium to bring a friend, right? Bring glucose across the membrane. So this is why we talk about all those membrane transport mechanisms way back in A&P1. It really, really is important for the big picture stuff that we're talking about now. Okay, any questions? All right. You thought you were done with all that cellular stuff, but nope, it comes back. Okay. So we've talked about a few of the structures. Let us start to add some physiology in here. So the blood flows, as we know, into the renal corpuscle, right? It was delivered to the kidney by the renal arteries. At the corticomedullary boundary, these smaller arteries here are going to branch off, delivering blood into the glomerulus via the afferent arterial. Okay. Filtration is going to happen within the glomerulus or the glomerular capillaries across/through what are called fenestrated capillaries. Okay, remember that fenestrated capillaries have those windows, those fenestra within them. And so this allows much bigger things to cross over the capillary membrane and into Bowman's capsule than would at, just like in your muscles or something. Okay. And then again, as we've discussed, the efferent arterial is going to drain the excess or drain blood out of this capillary bed and into the next capillary bed. Okay. And so I want to point out that the afferent arterial is usually much wider in diameter than the efferent arterial is. Okay, and so we've kind of talked about this concept before. In the lymph nodes, we have lots of different vessels coming into the lymph node, right? So essentially a huge volume of lymph coming into the lymph node, but only one single exit point out of that lymph node. And essentially what that does is it's like merging lanes of traffic. It holds up progress, right? That lymph is just like hanging out in the lymph node. What happens here, however, is that this is our blood circulation, right? So we have the pressure that is generated by the heart behind this blood. And so instead of just kind of like hanging out in the capillary bed, like lymph would hang out in a lymph node, instead, blood just keeps on coming into this capillary bed, pushing, pushing, pushing with all of the pressure of the heart, right? So what happens here is that there's lots of traffic coming into the capillary bed, one lane of traffic coming out of the capillary bed. And so instead of just hanging out in the capillary bed, we're going to get off at some exit ramps, right? So the additional pressure of the blood coming in through the afferent arterial is going to push blood, and a lot of it, across this membrane and into Bowman's capsule. Okay, so it's much easier to get off at the exit ramps than just to sit there and wait to merge into the one lane of traffic, right? Lots of traffic analogies, right? Because we all experience this all the time, right? So again, the larger diameter of the afferent arterial compared to the smaller diameter of the efferent arterial is going to increase the filtration, right? It's going to push more filtrate, more fluid with any solutes that happen to be in there across this membrane and into the tubule. And does that make sense? Is that okay? All right. So let's zoom in here just a little bit. Again, this is the afferent arterial. This is the efferent arterial. This is actually much wider in diameter. And as the previous slide shows, we can actually change the diameter of both of these arterioles to increase the pressure and therefore increase the filtration, or decrease the pressure and therefore decrease the filtration, right? So that's what's coming up next week. We're not going to talk about all those little details right now. All you need to be aware of right now is that the blood pressure is what drives the filtration process within the capsule, right? From the glomerulus into Bowman's capsule. And that is increased, and therefore we increase the filtration by having a really wide afferent arterial and a narrower efferent arterial. Okay, this is going to push filtrate contents of the blood across the glomerulus membrane first, right? So through the capillary bed membrane. Remember that these capillaries are fenestrated. They have these little holes in them, which allows larger substances to pass through. However, we still do not allow blood cells to pass over these capillary membranes, right? The only capillaries that allow the passage of red blood cells are the sinusoid capillaries, right? So the ones in the liver, right? The ones in the bone marrow, not the kidney, right? If you see blood in your urine, that's a problem. You are never supposed to have blood cells crossing over this membrane. Okay. All right. We also, have the Bowman's capsule membranes, right? So the visceral layer of Bowman's capsule, right? The ones that are actually on the surface of the capillary membranes, looks kind of wild, right? So there are not just normal old epithelial cells, but branching epithelial cells called podocytes, right? So these are like the feet cells with little fingers. And essentially, if you look really closely here, at a podocyte, you can see that there are these little cytoplasmic extensions that are literally interdigitating with, right? So weaving together with their neighboring cells. Okay. What this does is it leaves just little tiny slits in between these little extensions here that add a second layer, a second type of filtration, right? The gaps themselves are called filtration slits. Okay, and they're what allow these substances within the plasma to flow from the capillaries into the space of Bowman's capsule, okay, and ultimately get pushed through this next portion of the tubule. Okay, so again, the substances in the blood are going to be filtered. They're going to be pushed through the holes, the fenestrae of the capillary membrane, and through the filtration slits of Bowman's capsule, visceral layer of the membrane. Okay, and to be thorough, the outside, right, the parietal portion, the wall of Bowman's capsule is just made of simple squamous epithelium, right? It can be cheap, it can be simple. We don't really need it to do very much. We just need it to keep that filtrate inside. Okay. So here, just another perspective on what's happening here. Blood is filtered in the renal corpuscle from the blood, right, which is in here, across the fenestrated endothelium of Bowman, or of the capillaries, right, the glomerular capillaries. So these little holes are part of the capillary membrane, the basal lamina, and of course the podocytes, which have these little filtration slits between them. Okay, the filtration slits again only allow small-ish molecules to pass. Okay, so bigger than what you would get like in your skeletal muscles, for example, but still relatively small. All right, no big clunky proteins, no cells. Okay, and so this is the direction of our filtration. Right, so from the blood into the space of Bowman's capsule. Whatever can fit is going to pass over this very complex membrane. And essentially this produces a filtrate that has the same concentration of solutes, right? Not cells, but same concentration of solutes as the blood does. Okay. Questions? All right. Let's draw in what we know. Can you guys see the whiteboard here? Is it okay? I could draw back there if this is not going to work out, but I want to try it this way. All right, perfect. Thanks guys for the feedback. I always appreciate that. All right, so let's start here with a little boundary. Okay. This line is the corticomedulary boundary. Okay, so up here, this is the cortex, so the outer portion, and down here is the medulla. And again, that's going to become super important here. We know that blood is delivered via these arteries. At the corticomedullary boundary, we get this branching event, branching off with the afferent arterials. Right, so this is an afferent arterial. It is going to Bowman's capsule. I remember that the afferent arterial is actually much wider than the efferent arterial. So I'm going to draw this in a little bit bigger. Okay. So this is delivering oxygenated blood to Bowman's capsule, or to the glomerulus, that is. All right, so this big funky looking knot of capillaries is called the glomerulus. Okay, the capillary bed itself. Oops. Okay, so blood is coming in. There is the Bowman's capsule surrounding this, and it kind of acts like a little catcher's mitt. Okay, and filtration is going to be facilitated, encouraged, promoted by the blood pressure of the blood entering the glomerulus via the afferent arterial. So we're going to have, stuff, right? Salts and water and anything at all that is within the blood that is small enough to fit through these filtration slits entering into Bowman's capsule. That is this right here. Okay. Again, remember that the exiting blood vessel is going to be super skinny. And that actually promotes the filtration process. Efferent arterial. Okay, that's the efferent arterial. We'll come back to that in just a moment. Let's see. As I mentioned, the contents of the filtrate at this point are pretty much exactly the same as the contents of the blood, minus the really big stuff, right? So minus the cells, minus the big clunky proteins. We can talk about the concentration of like everything in these different fluids by saying collectively how many what are called milliosmoles are within these fluids, right? And so the actual numbers themselves aren't critically important. The units themselves aren't critically important, but I'm going to tell you that the concentration of solutes, of stuff inside the blood / inside the cortex of the kidney is going to be about 300 milliosmoles. Okay, and the reason that's important is because we are going to talk about diffusion, right, of water, of solutes across membranes. And so remember that stuff always flows down its concentration gradient. And so 300 is kind of the baseline here in the cortex. With that in mind, the filtrate at this point, right, that we have literally just made is also 300 milliosmoles, right? So whatever solutes were in the blood are now also in the filtrate. Okay, and so the filtrate entering into this next portion of the nephron is 300 milliosmoles. And what we're going to talk about on the next slide here is what's happening in this next portion of the tubule. Okay, note that this tubule is super squiggly, right? I didn't draw it that way just for the heck of it. It's super squiggly. Or a word for squiggly and not really in a straight line, not really straightforward, is convoluted. So this first portion of the renal tubule is called convoluted, right? There are actually two of these super squiggly portions of the nephron. This one happens to be closer to the renal corpuscle, so closer to Bowman's capsule and the glomerulus, and so we will call it the proximal convoluted tubule. Okay, and we can just identify that because it's closest to Bowman's capsule and it is super squiggly. Also, this proximal convoluted tubule is located exclusively within the cortex. Again, that's not on accident that I drew it that way. That is how it is. Okay. We can abbreviate this as PCT, proximal convoluted tubule. Please don't do that on a lab practical, but I'll be using that for brevity as we go through here. Okay, questions at this point? All right. As always, it'll come together. Okay, so the function of the proximal convoluted tubule is really to start pumping back lots and lots of that stuff that we just digested, we just got into our body through the digestive system. But the problem here is that there is no concentration gradient, right? We know that there is no passive movement of stuff unless there is a concentration gradient. And so the efferent arterial is skinnier than the afferent arterial because we want to have as much filtration as possible, right? We want to push as much stuff into the renal tubule as we possibly can. And we do that by, essentially giving the blood very little opportunity to leave the capillary bed, except through this right here, right? Through the filtration membrane. Right, it is literally easier for stuff in the blood to cross over through that filtration membrane into the tubule than it is to wait in line to get out via the efferent arterial. Right, so most of the volume of the blood in this capillary bed actually leaves the capillaries and enters into the PCT. Okay, the remaining blood, right, again, has the same concentration, and it is going to enter into this next capillary bed here. All right, and so it's going all over the place. All right, splitting into a capillary bed, weaving all over the tubule, this nephron here. Okay, I would like to zoom in on this interaction right here. Okay. All right, so what we have is the membrane of the tubule, right, which is made up of a bunch of cuboidal cells, right, simple cuboidal cells. So they all have little nuclei in here, one single layer of boxy little cells. All right, that is the proximal convoluted tubule, right, in this space here. So that's where the filtrate is as of now. We also have the capillary membrane, okay, which is super skinny, right? So essentially simple squamous epithelial tissue. Okay. So this is in our blood. Okay. In the middle here, what we have is something called interstitial fluid, which I've mentioned quite a few times at this point, but I understand that we haven't really gone into it that much. So this is just literally the fluid surrounding all of these different cells, all these different structures. Okay, so in the beginning of the PCT, we don't have a concentration gradient, right? And so the very first thing we need to do is generate a concentration gradient. And we happen to have these pumps that we all know and love, surely, that are super good at pumping sodium and potassium. All right, so I'm going to insert these little sodium-potassium pumps on the edge of these proximal convoluted tubule cells. What that's going to do is essentially pull sodium from the filtrate, push it out into the interstitial fluid, and ultimately it will diffuse into the blood. So again, the very first thing we do in the proximal convoluted tubule is we take sodium and we actively pump it across the PCT membrane and into the blood. Okay, so now we have a lower sodium concentration, sodium concentration within the filtrate than we do outside the PCT. As we know, water follows salt. Okay, so we have aquaporins in the membrane of the PCT as well. Okay, because water follows salt, right? Higher salt in the blood than there is in the PCT right now, water is going to flow down its concentration gradient through the aquaporins, following the salt into the blood. All right, so water is therefore reabsorbed. Okay, water is reabsorbed. And in fact, the overwhelming majority of the water that has just been pushed into Bowman's capsule from the glomerulus is going to be reabsorbed right here in the beginning. If we did not reabsorb all of this water, all of this salt, we would essentially pee out the entire volume of our blood in less than 10 minutes, right? So this is a huge deal. We really need to take all of that water and bring it back into our blood. Okay. Also, we don't want to forget about all those lovely things that we just digested and such. So things like glucose, amino acids, vitamins, right? All these lovely things that our digestive system just absorbed into our body, we don't want to just pee out. And so essentially what's happening here is that as the water diffuses out of the tubule and back into the blood, all of the stuff that's left behind, right, the glucose, the nutrients, the vitamins, everything, is going to get more and more concentrated, right? Same amount of nutrients in less water, right? So now the concentration of these guys is going to be huge as well. And so if we would then stick some glucose channels, for example, amino acid channels, for example, into these membranes as well, then all of these things can flow down their concentration gradient again into the interstitial fluid and ultimately into the blood. So really, the main function of the proximal convoluted tubule is to reabsorb stuff back out of the filtrate that we literally just made and put it back into the blood. So if we zoom in up here at the top again, right, salt, water, glucose, all these things are flowing through this tubule, and they are going to be pulled back into the blood here, right? So coming out of the filtrate back into the blood the entire length of this super wiggly section, right? And the fact that it's super wiggly and convoluted should indicate to you that we need a lot of surface area for these things to happen, right? Our structures don't wiggle for no reason. We always are increasing surface area whenever we see wiggles, okay? Questions about that? Does that make sense? Maybe? Oh, hopefully. And I'm gonna just minimize this a little bit. Okay. So let's go back to our PowerPoint here. All right, so a little bit prettier here. Filtrate enters into the proximal convoluted tubule. The very first thing that we need to do is generate a concentration gradient so we can reabsorb most of the filtrate. We do that first and foremost with the sodium potassium pumps on the basolateral side, so on the blood side of the tubule. We pump the sodium across, therefore creating a low concentration of sodium in the cell. Sodium then is going to diffuse down its gradient out of the filtrate and ultimately into the blood. All right, so that is primary active transport, literally pulling salt back into us. This gradient that is also kind of produced by this process is going to also drive the reabsorption of organic nutrients, right? So the glucose, the amino acids, etc. It's going to cause or lead to the reabsorption of lots of water. Water always follows salt. So if salt is coming out of the filtrate or the soon-to-be urine and into the blood, water is going to do the same thing as long as we have aquaporins, and we'll get to that here in just a moment. Okay. All right, so again, different way of showing this. If we take a section through the proximal convoluted tubule, we have the filtrate flowing through that tubule. So here's that fluid, and all of these things, glucose, water, ions, are all going to be reabsorbed back into the capillary via the generation of this concentration gradient of sodium, right? So that really does get the ball rolling here. Okay. In addition to reabsorption, so taking good stuff back into our body, we also have a lot of secretion, right? So stuff that was left over in the blood after the filtration process. And essentially we want to get rid of that if at all possible. And so we push these things into the filtrate as well. So things like urea, right? We're making urine here. And so putting urea into this waste makes a lot of sense. Creatinine, remember this is a breakdown, or it's a byproduct of breaking down creatine phosphate in your muscles. So that first burst of ready energy when you start contracting your muscles, that is leftover, or this is the leftover process from that. Uric acid, right? Remember we talked about this literally our first day of class together. This is a byproduct of breaking down nucleic acids. Okay. Also, potassium, right? If we are pumping sodium back into the body, we are also pumping potassium into the filtrate, right? Just because they are antiporters. If sodium is coming in, potassium has to go out of the body. Ammonia, different types of hormones, right? So when our endocrine organs secrete hormones, we don't want to produce this permanent change for the rest of our lives. Instead, the action of the hormone is only going to last as long as that hormone is circulating within the blood. Okay, and so the kidney is constantly filtering hormones out of the blood so that we can decide, are we done with this response or not? All right, so we're getting rid of hormones. If we want to continue that response, we need to keep dumping more hormones into the blood. Okay. Also, other things like drugs and toxins. The liver, of course, pulls them out of the blood and like changes them a little bit, but we need to physically get them out of the body somehow. And so the kidneys are going to help with that. Also, any excess nutrients. So if you have a super sugary meal, or like, you know, Halloween a few weeks ago, you ate an entire bag of Halloween candy, that is way too much sugar for your liver to process and your tissues to take in, especially if you have an issue with your pancreas, such as diabetes. And so some of that sugar will end up in your urine, right? We don't want to have too high of a blood sugar. Okay. And finally, your kidneys, again, are going to help with pH balance. And so they can either push hydrogen into the filtrate or keep it in your body, etc. Right? We'll talk about that again next week. But here is where we really start to play with this pH balance. Do we need more hydrogen to pull the pH down, or do we need more bicarbonate to pull the pH up? Okay. And so this is a summary of all these different processes within the PCT. The PCT really functions primarily in reabsorption. We reabsorb about 108 liters of water every single day via our proximal convoluted tubule. And so again, if the PCT was unable to reabsorb all of this water, we would literally pee out the entire volume of our blood really quickly. Okay, so that's the PCT. Let's quickly do a poll to see what we know so far. A typical renal blood flow is about what percent? So what? Percentage of cardiac output under resting conditions. Thanks, Guy. This is good news. All right, fantastic. You're absolutely right. 25%, right? A quarter of your blood volume is going to be filtered through your kidneys every single minute. All right. Sorry. Here, another question. The glomerulus is what? The expanded portion of a nephron, so actually holding the filtrate itself, a knot of capillaries within the renal corpuscle, the source of EPO, attached to the collecting duct, or a horseshoe-shaped segment of the nephron. All right, seems like we're good with that as well. So the glomerulus is a knot of capillaries within the corpuscle. What is attached to the collecting duct is the distal convoluted tubules. So we are getting there as well. All right. And finally, I can't even see the question. The process of filtration is driven by what? Sorry, I'm clicking all the wrong things. Okay, I think you can see that now. So the process of filtration is driven by active transport, blood osmotic pressure, blood hydrostatic pressure, renal pumping, or solvent drag. All right, so keep in mind that filtration is one of 3 processes occurring within the nephron: filtration, reabsorption, and secretion. All right, this one's a little trickier, right? So here is what you guys said. Let me point out that filtration is what's happening. All right, here is the glomerulus, the capillaries, and here is Bowman's capsule. Filtration is what's happening from the blood into the into Bowman's capsule. And so this is actually driven by the pressure of the blood. Okay. So active transport is what is going to drive reabsorption from the filtrate back into the blood. So those of you who have said active transport, that is an important process here, but that is more for reabsorption. Okay. And later we'll see secretion as well. So something to do with the blood, is it blood osmotic pressure or is it blood hydrostatic pressure? We are going to be talking a lot more about this next week, but we know enough to answer this question at this point. Remember that osmotic pressure is essentially the pull of water and filtrate here by solutes, right? So this is by solutes. So that would essentially be saying that there's lots more solutes in here than there is in the blood, okay? Hydrostatic pressure is the pressure literally of the fluid itself, the physical pressure or the blood pressure, okay? So hydrostatic pressure is actually always pushing out of the capillaries in the glomerulus. And so the correct answer here is actually C. Okay, and so that one is a little bit trickier, absolutely. But we have talked about osmotic versus hydrostatic pressure long ago in the first section of this class when we talked about the normal capillary beds. And so those concepts are going to come back. So I encourage you to look at those again. Okay. All right. The next thing that's going to happen within the nephron is the filtrate is going to flow from the proximal convoluted tubule into the loop of Henle. And so we're going to finish up today with our little diagram here. We'll talk about the physiology together on Monday. But for now, let's finish this drawing and then we'll come back to that later. All right, so from the proximal convoluted tubule, the remaining filtrate, whatever is left over that hasn't been reabsorbed back into the blood, is going to enter into the loop of Henle. Okay, so let me draw this in here for you first. The loop of Henle is going to descend or allow the filtrate to descend into the medulla, more or less straight down. Right, and there is a fatter portion here that is important, right? And so again, this structure is going to descend down into the medulla. This half of the loop of Henle, which is also called, right, wait for it, Henle's loop or the Nephron loop. Okay, so many different names for the same thing, but in general, it's the loop of Henle. There are two different limbs of this loop of Henle. The first one is the descending limb. Okay. So the descending limb is allowing the filtrate, or it's bringing the filtrate down deeper and deeper and deeper into the medulla. At a certain point in the medulla, and it varies depending on which one we're talking about here, the loop of Henle is literally going to loop and come back on up. And again, there's a skinny portion and there's a fat portion of this loop. Okay. This is the descending limb over here on the left, allowing filtrate to go down. One might assume that on this side it is called the ascending limb of the loop of Henle. And again, this is what allows the filtrate to ascend back up into the cortex. Okay, something that's going to become very important on Monday, right, when we talk about this again, is the fact that as you go deeper and deeper and deeper into the medulla, the concentration of the fluid surrounding these loops of Henle is going to get higher, right? So maybe here goes from 300 to maybe 500 milliosmoles, right? Deeper yet, maybe now it's at 800 milliosmoles. Okay, and all the way down here at the bottom, the very deepest, 1200 milliosmoles. Okay, and so this is the same thing on both sides. These nephrons are just suspended within this super concentrated fluid. And that is going to be really important in concentrating the urine, right? Reabsorbing even more water than we already have so far. Okay. When the ascending limb of the loop of Henle crosses back over into the cortex, it's going to get skinny again, and it's also going to get wavy. Okay. This portion of the nephron is called the distal convoluted tubule, okay, or the DCT. Okay, on a practical, spell it out for me, but I will use the abbreviations. Okay, in the distal convoluted tubule, as we'll see in our next class together, we are going to make our last ditch efforts to modify the filtrate and modify the blood. Okay, this is where we purposely say, okay, we have too much calcium in the blood, like let's put more into the filtrate. You know, we have too many of this other thing, let's put it into the filtrate. We don't have enough of bicarbonate, for example, so let's pull some more bicarbonate out of the filtrate back into the blood, right? So all of those very specific modifications for the specific needs of the body is going to happen in the distal convoluted tubule. Also, if you have high blood pressure or low blood pressure, this is really the place where we're going to modify that. Okay, the distal convoluted tubule is going to dump the filtrate, whatever's left, whatever we have secreted and not reabsorbed is going to get dumped into this final structure here. This final structure is called the collecting duct. Okay, and that is going to take the urine, right? At this point, it's urine, down and out of the kidney and ultimately to the urinary bladder. Okay, there is a tiny little bit more modification occurring here. And again, this is super selective, right? We can put channels in the collecting duct to reabsorb more water or less water or more salt, less salt, etc., depending on the needs of the body at the time. Okay, so that is where we're going to go in our next class together. I'm going to leave this up for a minute so that you guys can finish drawing it out. But of course, this is being recorded, so you are welcome to look at it again online. Are there any questions at this point?